Hi everybody. In this video, I'm going to try to connect a bunch of stuff that we've talked about in this unit, everything from how cells grow and divide, to how you use your DNA, to how you make proteins. Now, the centerpiece of this video is going to be the cell division cycle as drawn right here. Now, the first thing I want you to realize, as you might already know, most of the cells in your body are not in the cell division cycle. If you're a neuron cell of the brain or a muscle cell of the heart, you're generally not growing and dividing. You are in this state that you can see listed called G0. Now, in certain cases, you might get pushed into the cell division cycle, but most cells in your body are not. The cells that are in the cell division cycle are the ones that you typically call stem cells, the ones that are constantly at some rate growing and dividing and then becoming specialized or differentiated cells eventually. So I'm going to draw you one of these cells. So this is going to be some sort of a stem cell that has not become a specific cell type yet, and I'm going to draw in the chromosomes for you. Now, to make this easier, I'm not drawing in all the chromosomes. In a human cell, there are 23 pairs of chromosomes. You don't want me to draw that. I'm going to make this simple. I'm going to draw two. So here are two chromosomes in a cell that is entering G1. Now, to do the cell division cycle completely and make two cells that are identical to the original, you have to kind of do two or three things. One is typically to just grow, maybe double your size before then copying your chromosomes, ripping them apart, and then cutting the cell in half. So here's our first step. Let's imagine this cell has to grow. That's G1. G doesn't actually stand for growth, but good enough for us for today. So G1, growth. So the next step is going to be S phase. S stands for synthesis. This is where you're going to copy the chromosomes. If you're going to make two cells, you better have all the chromosomes for those two cells. So here's the next view of the cell. I've zoomed in just to make things easier to see. The cell wouldn't have gotten this big. All right, so here we are. We are in DNA synthesis phase, S phase, where we copy the chromosomes so we can separate them into two cells. As you can see, I've already drawn one of the chromosomes, the shorter one, starting to get unwound. As we've modeled in class before, there is an enzyme that pulls apart the two strands. It happens to be helicase. And then there are going to be machines that match nucleotides to those two strands that have been separated. These are called DNA polymerases, and here they go. There's one, and there's the other one doing its job. Let's take a look at the next chromosome. This one's a larger chromosome, and let's imagine it's getting pulled apart again by some helicases. Now, on each of these strands, you're going to have polymerases copying, or in reality, matching that strand, and then making the other strand that is appropriate. So here's all of that done. And you can see in the second chromosome, I've actually sort of curled them back up into double helices. The first one, I left them flat to make it simpler to understand. Okay, so what's actually happening with these chromosomes? So I'm going to draw you a sample bit of a chromosome. There's one strand in one direction. There's the other strand anti-parallel. Now, if you're going to replicate this chromosome, if you're going to copy it in S phase, first thing you have to do, just like I suggested, is pull those two strands apart. So there we are, separated. And then on top of each of these two strands, we're going to put the matching nucleotides. So across from the G is a C, across from the A is a T, etc. That's one of your new strands. Here's the other new strand. And in some cases, you'll make a mistake, and I've drawn one in there, and that's the red T. And you can call that a mutation. When you incorporate the wrong letter or a missing letter or an additional nucleotide, that is called a mutation. Okay, you will now notice that each of these chromosomes, or partial chromosomes, the double helices on each side, each one of them is half of an old piece of DNA. So one strand that's old, that's the dark purple, and one that's new. That happens to be called semi-conservative replication. And if you want to know that, you're welcome to know that word. And these identical copies of the chromosomes happen to be called sister chromatids typically. That's not essential vocab for this unit, but you'll probably see it in the next unit. All right, next. So we've now copied our chromosome. We have grown, that's G1. We've copied our chromosome, that's S phase. And now we have to transition closer to the point where we're going to pull apart those copies and cut the cell in half. Now, G2, S phase is done, so let's look at the chromosomes. Back in G0 or G1, we had 2 chromosomes. Now we should have 2 of each of those, so a total of 4. Now, to compare that to us, if we were talking about human cells, we would have gone from 46 chromosomes here to, I guess, 92 things that you could call chromosomes. All right, next. Our last phase of the cell division cycle is going to be M phase. And M stands for mitosis, or mitotic phase. And in this, and let me zoom in pretty far here to make it nice and big, we're going to take those chromosomes, make them nice and small so we can sort of pull them apart without them getting tangled, and then yank them apart. And that's what I've drawn here. The 2 longer chromosomes getting pulled apart from each other, the 2 smaller ones getting pulled apart from each other, and some cords of protein pulling on them in green. There are kind of 3 steps, as I mentioned. The first is to condense the chromosomes so they don't get tangled. I haven't really drawn that here because at no point have I shown them at the full length, so I can't really show them much smaller than I already have. But again, you're going to condense them, make them nice and small so that they're easy to pull apart without them getting tangled. Then you're going to pull the 2 identical copies apart, so both cells, after you cut the cell in half, will have a full set of chromosomes, and then split the cell. This process is called cytokinesis. Here we go. And you should also reform nuclei around those chromosomes. Okay, now what this leads us to next is the fact that in some cases, cells might do this a little bit too much. This whole division, grow and division thing. So what I'm going to draw for you is a part of the body. Let's imagine this is the lining of your stomach or something of that nature, or your intestines. And so we have an epithelial layer of cells, that's the top layer in brown, facing, the inside of your stomach, for example, and then a connective tissue below. Now, most of these cells, as I mentioned before, are in G0. Most of them are not growing and dividing. Most of them are just doing their job, either sending out digestive enzymes or absorbing nutrition or whatever it is that they do in this case. So again, let's imagine this is the stomach, but a couple of these cells once in a while will get worn out and die. And they might, leave a gap. Now, this is not exactly how it really works. I'm just trying to simplify this. You would not have a hole in your stomach on a regular basis. But this should be easier to understand than the reality of it. So let's imagine that a couple of the cells have worn out and died. The cells next to them will want to, or will get some sort of signal suggesting that they need to go from G0 and start to grow. They need to get into the cell division cycle, start to grow and divide to fill in that gap. So let's say they enter G1 and then continue the cell division cycle. They're going to get bigger, copy their chromosomes, and then pull them apart, divide the cells in half. And now we've replaced those 2 cells that we were missing. Occasionally, though, some of these cells will go a little out of control. If they're damaged, if their DNA, for example, has been mutated in specific ways, or maybe you even inherited some mutations from your family, these cells might start to divide a little bit too fast. Maybe they go through the cell division cycle too quickly or too often, or maybe they just go into the cycle when they're not supposed to. So you might get a few extra cells. This is not especially dangerous on its own. If you get a bunch of extra cells, this is also not especially dangerous on its own. There is a word for a big old mass of cells like this. Maybe you've got a clump on the inside of your intestines and your colon. You could call this a colon polyp. You might have a little extra mass of cells like this on the outside of your skin. This is technically a tumor, but it's not necessarily dangerous. This is the kind of small little mass that you could burn off or cut off or cinch off without too much difficulty or danger. The danger comes about, though, when cells start to invade other locations, when cells from the tumor might start going from the lining and going deeper into your body. At this point, we have invasion, and this is now a cancer. So I just want you to see that the cell division cycle has to be really well controlled. If it goes out of control, you end up with tumors which can become invasive and become cancers. All right. So what we have not talked about yet is what our cells normally doing. So back to G0. So we've spent this entire video talking about how you go into the cell cycle and how you make your way through it and what was happening specifically to the DNA. What we have not covered is what is happening to the DNA or with the DNA in G0. So let's zoom in really far. So I'm going to have to make this way bigger than it normally would to be to scale. And I'm going to draw you, again, the same 2 chromosomes that you saw in the G1 cell, just zoomed in. All right, here's the short chromosome, but the one we're going to focus on is the large chromosome. If you are a cell in G0, you have to make proteins to do your job. If you are a muscle cell, you're probably making motor proteins so you can contract. If you are a cell that lives in a tendon, you're making collagen protein to strengthen the tendon. So here is one chromosome, and a couple genes are partially unwound. And I've given them just numbers that I've made up. These genes are being read so we can make proteins. So you're going to have 2 strands being separated. One is the strand that you read, that's the template, and one is the strand that actually has the recipe, that's called the coding strand. We're going to do a process called transcription to make a copy of that recipe. Here are the enzymes that do it. These are called RNA polymerases, and they do the transcription, and we're going to make a match to the template strand, so the copy of the recipe is shown right here. This is your mRNA. It's going to get processed and then exported out of the nucleus, and that is where we're going to do the translation out in the cytoplasm to make the proteins. Again, if you're in G0, you're just doing your job. You're making the proteins you need to do your job, but if you get shunted into the cell cycle, you kind of put that to a stop. You focus on, I got to crop my chromosomes, I got to divide those chromosomes into 2 piles, I got to cut the cell in half. But in G0, your chromosomes are decondensed, they're accessible to you, and you get to make the proteins that help you do your job. All right, folks, feel free to rewind, rewatch parts that are essential to you. I hope that helped bring it all together. See you next time.